I am Dr. R. R. Sanap, Head and Associate Professor, Department of Botany, Rao Bahadur Narayan Rao Boravke College, Srampur. Today, I am going to deliver the lecture on the reproduction in Pinus. So, it is the part of syllabus of TYBSc Botany, and paper is 4th, that is BO 334. Title of the paper is the Spermatophyta and Paleobotany. So in that, the gymnosperms, so this Pinus, it is a part of gymnosperm, and in this life cycle of Pinus, so I am going to deliver the lecture on the reproduction in Pinus. So this Pinus, if we see its reproduction, so it in the asexual reproduction, the reproductive structures, they are developed on the plants, and these reproductive structures, that is the male and female cones, they are developed on the same plant but on the different branches. So the Pinus, it is a monoecious plant. And that's why, as it is the monoecious, male and female cones develops on the same plant but on the different branches. So if we see the structure of male cone, so this male cone, it develops on the dwarf branches so many male cones, they develops in group or in cluster, and they develops on the dwarf shoot or the dwarf branches. So this here, this is the dwarf shoot, and this is the main shoot or main stem. And here on this dwarf shoot, here these male cones are developed. So these are the male cones, and these male cones, they are found in the cluster, and they get covered by the presence of the these needles, that is the Pinus needle, that is the leaves. So these are the Pinus needle, that is the leaves of the Pinus, and this terminal portion of this dwarf shoot, it ends into such type of this male cones. So thus these are the many male cones found in the cluster. So if we see the structure, internal structure or the LS of the male cone, then this Pinus, it shows the presence of the central axis. So here, this is the central axis, and on this central axis, here many microsporophiles are arranged. So this is the central axis, and on that central axis, here the many microsporophiles are arranged. And if we see this microsporophiles, so they get spirally arranged on this elongated axis. So the older one, they are present at the base, younger one, they are present at the apex. So each this microsporophyll, it shows the presence of the flattened structure, and it get covered by these scales. So these are the many microsporangia we get arranged or in the acrophytal succession. It is older at the base, younger are at the apex. So it get covered by these Bragg scales. This is the central axis. The central axis, it is elongated, and that elongated central axis, many these microsporophylls are arranged. So these are the microsporophylls. So these are the mini microsporophylls. They show the presence of these appendages, and at its base here many microsporangia are arranged, and this microsporangia, as the name indicates, they show the presence of the many microspores, or they are also called as the pollen grain. So thus here this microsporangia, they show the presence of the many microspores. So these are the microsporangia, and in this microsporangia here the many microspores are produced. So these are the microspores, or they are also called as the pollen grains. So microspores or pollen grains. So thus here, if we see this entire cone, then it is actually they are found in the bunch or groups. If we enlarge this, it shows the presence of the short stalk central axis, and on that central axis many these microsporophylls are arranged, and each this microsporophyll shows the presence of these Bragg scales, and at the base of it here it shows the presence of the many microsporangia, and this microsporangia, they produces the many microspores. So if we see this single microsporangium, then its structure it look like this. So it shows the presence of the elongated conical structure. It is known as the apophysis. So thus here, this is the apophysis. And below it, it shows the presence of the two pendant, this microsporangia. So these are the microsporangia, which are present in the pendant condition, get attached towards the lower side of this apophysis, and it shows the presence of the many microspores. So thus here many microspores or pollen grains are developed in this microsporangia. So these are the microspores, or we may call it as the pollen grains, microspores or pollen grains. So thus it is having a short stalk. With the help of that stalk, it get attached to the central axis of the cone. So thus here this is about the single microsporangium. Now when this male cones, they becomes mature, later on this male cones, they becomes dry, and then later on each this microsporangium, which is get covered by this apophysis in the microsporophyll, it dehisces. And after the dehiscence of this microsporangia, so here the longitudinal slit is developed, and through this slit all the pollen grains then later on they comes outside. All the pollen grains they comes outside after the dehiscence of this microsporangia, and later on these microspores or pollen grains, with the help of the dry weather, they get transferred or get dispersed from this male cone towards the female cone, and thus it is known as the pollination. So in case of the pinus, the pollination takes place with the help of wind, that is the anemophilous pollination. As the flowers they are not attractive, that is the male and female cones, they are not attractive, with the help of wind the pollination takes place. Now if we see each structure of each this pollen grain, then this pollen grain, it shows the presence of the structure like this. So it shows the presence of the two walls. So this outer wall is called as the exine, and inner wall is called as the entine. So thus here this is the exine. Exine, it is somewhat tough layer of this pollen grain, and inner side to this it encloses a thin layer, it is known as the entine. So this is the entine. Entine is thin membrane, and it encloses here the cytoplasm. So this is the cytoplasm, and there is a presence of the nucleus. So the nucleus, it is present in the center. Single nucleus is present, and this pollen grain, it get covered by the two wings. It get covered, or it show the presence of the two wings. So these are the two wings, and with the help of these wings, here these pollen grains, they get float in the wind current. So when this sporangia they dehisces, all the pollen grain they get liberated, and with the help of wind, these pollen grains they get dispersed or get pollinate. So during this the pollination, these wings, they are playing very important role. They act as like a parachute, and from the this male cone, then they get transferred towards the female cone, and thus the pollination is taking place. So thus here this is the structure of pollen grain, showing the main body, that is having the exine, entine, cytoplasm, nucleus, and the presence of these two wings, and with the help of wings they get transferred from the male cone towards the female cone. So thus here this is about the structure of the male cone. So it shows the presence of, this is the internal structure, that is alias of male cone, then the microsporophyll, then the microsporangia, and in the microsporangia many microspores or pollen grains are developed. So thus here it is about the male cone of the Pinus. So here I am going to conclude. Thank you very much.